Welcome back, Year 12s. Let's get into a big video looking at the TRP Operon. So we'll do a quick recap of the last video. All right, we looked at translation specifically. And again, if you don't understand transcription, RNA processing, translation, this is not the video for you. Go back to the other ones. But if you do, this is the next topic we're covering. So translation, as you can see, is the creation of the polypeptide up here, which is our protein, all right, through regular amino acids being joined together through peptide bonds, okay? That all happens in the ribosome here, as you can see, through the messenger RNA. All right, that's just saying the same thing. The ribosome binds and then reads the messenger RNA, and then the anticodons of the tRNA come along, and they essentially bring each individual amino acid. All right, corresponding amino acids are joined with one another, okay, through a condensation reaction to create that peptide bond. Multiple of those will then create your polypeptide. All right, and this is your codon chart that you should be able to read by now. Alrighty, so gene regulation. So we're looking at the TRP Operon. And obviously TRP, or tryptophan, is one of the amino acids, okay? And that's where the regulation comes from. It needs to be regulated, all right? In particular, organisms, and in the case of what we're looking at prokaryotic organisms. Cool, so that's your dot points. Awesome. So we'll take a step back and we'll look back at our genes, okay? And just remembering our prokaryotic genes are, of course, circular chromosomes. All right, they're not linear like our eukaryotes. This blue section here are, of course, our structural genes or our genes. Normally that would be introns and exons, but in this case it's no introns. So only expressed genes are going to be here, okay? They have this extra region called the operator region up here, which is going to be important soon, all right? And essentially a cluster of genes together, which no introns means they're all clustered up. That's what we call an operon, okay? So we have our TRP operon, which is just the operon, so the cluster of genes that helps create tryptophan, okay? And essentially this needs to be regulated. All right, what else do we look at for prokaryotic genes? mRNA has no methyl G cap, okay? So as you can see here, no 5' methyl G cap, no 3' poly A tail, as the, well, in this case, the ribosome is doing translation, but transcription has also occurred before that, okay? And translation is concurrent with transcription. So what does that mean? It just happens at the same time. So realistically, if I was to quickly draw something on here, there would be another protein running here. And what would that be? That's right, RNA polymerase. Okay, and that's essentially what's doing the transcribing. Just before the ribosome then does the transcription. That's right, the translation. Okay. Okay, so that brings us to gene regulation. So we have these regulatory genes that code for proteins that influence the expression of structural genes. Okay. This prevents the over or under expression of a particular protein. Really, that should be amino acid in this case for what we're looking at, and allows the cell to adapt to its needs. Okay, so amino acids such as tryptophan can be very energy expensive. Okay, so if you think of any prokaryote, E. coli, Escherichia coli, okay, or Staphylococcus aureus, something like that, any bacterial cell, they need to create tryptophan, okay? And in this case, they don't want to create too much tryptophan because it's so energy expensive for the actual bacterium or actual prokaryote, okay? So it wants to look for efficiency. That's why we have regulation of these processes. So in our prokaryotes, such as bacteria, we have this region here, okay, which we call the operon. All right, it has our structural genes in the DNA sequence or RNA sequence, okay, but it also has this promoter region and an operator region. So that's what we're really looking at, the operon here. But also, there's something to note is that there's upstream genes too. So that's a regulatory gene and also a promoter, okay, but mainly this regulatory gene. That's going to come back later, okay. But also, we're really specifically looking at the operon here. So why tryptophan as opposed to other amino acids such as arginine or methionine? All right, tryptophan is the rarest of the 20 essential amino acids. All right, there's more amino acids than that. We've seen that on the codon chart. But there's 20 really essential ones, and tryptophan is the rarest. Okay, so how do we get it? As eukaryotes, we really just get it from our highly protein-dense bioavailable foods. That's your meats, your fish, your eggs, anything like that. Okay. And why does it need to be regulated? Because it's so energetically expensive, as I said. When a prokaryote needs to make this, it uses a lot of energy in the cell. So the TRP Operon, here we are. We're looking at the main picture of it here. All right, it consists of a few different things. So firstly, you have your RNA polymerase. That's obviously going to what? You should be able to answer this by now. Correct. Transcribe the genes. Okay, and that's sitting on the promoter region. Okay, you also have this operator region, which is important. You have the leader region, TRPL, and that comes into play when we look at attenuation. All right, right now we're just looking at repression. And then we have the structural genes. So all these genes are required. So TRPE, D, C, B, and A. Ironically, it's backwards. Really, it should be A, B, C, D, E, but I'm sure there's a great reason for that. Okay. They are used to create the tryptophan. They all make different proteins, as you can see, because they're different types of genes. But they're all in this one operon. And lastly, we have the trailer region. Obviously, that has the stop codons at the end. Cool. What else would there be? If we look at the promoter again, if you go upstream, keep going upstream, you'll obviously find that regulatory gene, which then, we call TRPR. Okay, but that isn't shown here. All right, so again, these proteins will encode for different, these genes will encode for different enzymes, which are proteins, which are used in making the tryptophan, so tryptophan synthesis. Okay, so we're looking at a lot here, but there's nothing to worry about. So several different polypeptides created by the structural genes are a part of the operon. Okay, so TRPE, as you can see here, the light blue, makes the light blue polypeptide, which we mentioned as enzymes before. Well, the purple one makes the purple protein. Okay, TRPB makes the yellow protein, etc. All right, and they're just all a part of this process of changing one molecule of charismate into a molecule of tryptophan. This is why it's so energetically expensive. Think about the creation of 5 different proteins, and then the use of those proteins as enzymes to catalyze a reaction to essentially make one amino acid that could just be a part of TRPA, in theory. Okay, so that costs a lot to make tryptophan. All right, so here's the process here, and you do not need to know this. Okay, this is not a part of the VCAR dot points, but it is interesting to understand so that you can, understand the repression model of the TRP Operon better. So we have this readily available molecule, charismate. Okay, these two enzymes work together to make anthranilate from that. Okay, then you have the crazy purple one, again, with a silly name. All right, and then that makes another molecule called indole-3-glycerol phosphate. Okay, then we have the 2 yellow ones working together again as a quaternary structure protein or enzyme in this case. And that finally turns indole-3-glycerol phosphate into tryptophan. And there you go. You have one single amino acid tryptophan after you made all these things. That's very energetically expensive. And again, you don't need to know any of these names or the process that occurs here, but that is the process. Okay. You do need to know what the structural genes do. They are enzymes that help turn charismate into tryptophan. Okay. And so we have gene regulation by repression here. Okay. So TRP Operon repression model. And this occurs when there is enough tryptophan within the cell. Okay. So there's enough tryptophan floating around, free floating as amino acids do within the cell cytoplasm or cytosol. All right. If it's floating around and it doesn't need to be created, that would be a waste of energy to create it. So in this case, you have this repressor protein. And where did that come from? Where does this protein come from? That comes from upstream TRP regulatory gene. Okay, the TRPR gene essentially codes for this repressor protein. This repressor protein. Okay, as you can see, it has 2 slots, perfect for tryptophan to fit into. Okay, and that connects to the operator region of our gigantic strand, okay, of nucleic acid here. And essentially that connects to that operator region if it has changed its shape. Okay, so as you can see here, it doesn't have the little sticky up things, okay, but it does here in this example. All right, and what's the difference? There's a lot or enough tryptophan within the cell, free floating, so this repressor protein then attaches its binding sites to the free floating tryptophan. All right, and there you go. The protein changes shape. So then it can bind to the operator region. And guess what happens to old mate RNA polymerase here? It gets blocked. It can no longer run down the strand. If RNA polymerase cannot run down the DNA strand here, what isn't occurring? That's right. If it can't run down the DNA strand, transcription doesn't occur. No transcription means no translation, means no structural genes. We have no structural genes, that means no tryptophan will be created. Okay, and so therefore it represses the creation of tryptophan. It will never stop it, it will never change it to 0, but it will definitely significantly lower it. Okay, and this is where the students often get confused. Okay. All right, and this is our last thing we're going to look at. Again, this is the same TRP Operon with the same repressor protein, just shown in a different way. Okay, so this is putting it all together. Should be able to understand this, at least to a reasonable extent now. Cool. So if we just look at our DNA strand, found in our prokaryote, all the way upstream, which is represented by these lines, is our TRPR regulatory gene, which will then create our repressor protein. So that's then instantly transcribed, translated. Okay? Get to the promoter region, get to the operator region. All right? And as we saw before, you would have seen RNA polymerase just sitting here. That's sort of drawn in the green there. Okay? And normally that would just run straight down the strand. Boom. Okay? Starts to transcribe it, and then therefore it gets translated as well because they are concurrent, they happen together, and bang, you get your structural genes. And if you get all of these, tick, 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 that means charismate gets turned into tryptophan. Okay, and that's what we want. All right, or in most cases, that's what we don't want because that's a waste of energy. Okay, so the RNA polymerase would have run down the strand, but it didn't because it's been blocked. Okay, and it's now been blocked by the repressor protein, which sits on the operator region. Okay, as you can see here. All right, because there's enough tryptophan in the cell cytosol cytoplasm, so it goes into the binding sites, changes the shape, and then blocks that green RNA polymerase from transcribing. If it can't transcribe, it will never translate. Okay? And that's about it for the TRP Operon via repression. Okay? In the next class, we're going to look at TRP Operon via attenuation, which uses the TRPL or leader region here, as well as the attenuator region. It's a little bit different. Okay? Again, like, comment, subscribe. If you have any questions, ask me. All right, Mr. D out.